Hello, my name is Gwendolyn Squires. I am a McNair Scholar and a member of the Honors College at Sam Houston State University. My mentor is Temi Salami, and my research is on emotional broadcasting in high-functioning autism. And first, I'd like to go over an overview of my research. We're going to today discuss what autism spectrum disorder is, what microexpressions are, how microexpressions are related to autism, different neurology of autism, the gaps in the application of microexpression studies for neurological differences in autism spectrum disorder, the methods that I used for my research, and what the analysis of the data showed, and the future directions that we could take with research. So first, let's go over what autism is. The DSM says that it is individuals who suffer from restricted repetitive behaviors, symptoms present during early childhood development. Symptoms can cause clinically significant impairments. And what we're going to specifically focus on, persistent deficits in social communication and interaction. Abnormalities in eye contact and body language is mentioned, up to a total lack of facial expressions and nonverbal communication. And I want you to kind of keep in the back of your mind why this could be occurring and affecting individuals. We're going to look at what microexpressions are now and how they're being studied. They were first noted by Darwin, so a long time ago, and a man named Paul Ekman picked up the research where he noted a blueprint for people that emotions could definitely be read on the face. And those emotions, there are 7 of them, are universal. It doesn't matter if you were born blind, you will still show these same 7 emotions. They are disgust, anger, fear, sadness, happiness, surprise, and contempt. Now, he studied this in the terms of deception, and Matsumoto picked it up the same way, but he asked a very important question. Matsumoto looked for evidence that there was valuable applications in training to be able to read microexpressions of emotion. And he found that there was, that emotions can be trained, and being able to read someone's face and the muscles on your face and how they broadcast that emotion can definitely be read. We take it another step further to kind of look inside the brain at what's going on and how the social reciprocity happens. And Ariki did a study where he looked at mirror neurons in the brain, and he specifically looked at monkeys. And he was able to note that the ability to read and mimic behavior is involved with monkeys and humans. And it has been studied that social interaction with mirror neurons can sometimes be broken, even in clinical populations of autism spectrum disorder. Shen also noted that the duration is important. So a macro expression is 1 second or longer. It's if I'm smiling at you for a longer period of time and I hold that expression, I'm going to be read as being more genuine, whereas micro expressions are tied in the research to deceit. So how have microexpressions been related to autism then, if it was geared towards deception? Well, they have come up with apps that can capture your arousal levels. They'll take selfies of you after you've had certain feelings, either physiologically or your heart rate racing, and it will kind of train you in how you're feeling with your emotions, and also to be able to read the emotions in others by showing you pictures and impressions. So it's a feedback training tool, but it's only been moderately helpful. Now, Clark noted that children with ASD do show atypical facial reactions in others, and the mechanisms still remain unclear. We don't fully understand why. Now, there's new technology that can track levels of variability throughout the body. Torres is the researcher who has been studying this. And this is good news for us because it shows that movement is measurable. Even small movements of all the muscles in our face, which have a lot of muscles, will be able to show what we're really feeling inside. Now, if your brain is working one way and it's been identified universally that that's how it looks on your body, then that's how we've come up with universal emotions. But no one has looked at the fact that individuals with autism have a different neurological basis for their behavior. This has been shown in prosopagnosia, where people have face blindness, and we still don't understand the mechanisms for that. Synesthesia is the mixing of 2 different senses, where you can hear something that you see. Or when you're listening to music, you will see color. Those are the blending of senses, which are more disproportionately part of the autism community. Now, there is no, Williams and Fan went back and forth, and there was really no consensus as to if this was the imitation neurons involved or the motor neurons. We just don't understand what's going on in the brain. But it seemed to me that we were making a big leap in understanding emotions and the micro expressions, because honestly, we didn't understand what's going on in the brain. So how could we apply a universal model to people who are neurodiverse? So I formed my own methodology to do an experiment where we looked at participants. 10 were autistic and 10 were neurotypical, and they took part in a Zoom video recorded interview where they looked at a slideshow of images. Now we focus specifically on happiness. Genuine happiness reaches the eyes right here. You smile, there's some muscles that run along here. You have cheek raiser muscles. If it's genuine, it reaches your eyes. And we kind of use this model from the Paul Ekman group, the facial action coding system. However, I couldn't just look at one emotion. I needed to be able to contrast two emotions to make sure that people were actually being able to convey emotion. So we took happiness and sadness, and they looked at 16 illustrated cards. A few of them were baseline photos that the participants showed us themselves. So we would know that we really were looking at on their face was something genuine, something that they found happy or sad. And we standardized the order and sequence in which we showed it to them. Now, what we were really hypothesizing was that this rigidity of expression, this stoicism, is a totally misunderstood emotional expression for autism spectrum disorder, that they have a great deal of emotion, but that it may just not be the neurotypical way of broadcasting it. We showed that there would be a marked difference between how people with autism express their emotions and their neurotypical counterparts. Individuals with autism do not have a restricted emotional expression, but instead have a different microexpression language that creates translational issues during communication and social situations. So we asked if there was a recognizable pattern among how autistic participants broadcast their microexpressions of happiness or sadness, and if there was no correlation whatsoever. The data was interesting. So we decided that the best tools for statistical analysis would be a 2-sample chi-square with cross-tabulation for nominal data, followed by multivariate Kruskal-Wallis H-test, which kind of replaced the repeated ANOVAs that we thought we would be using. There are multiple dependent variables, and so we weren't able to use the repeated ANOVAs. We also used the Mann-Whitney U-test, as you can see, for ordinal data corrected with Bonferroni. So on the surface, we looked at the type, duration, and intensity that someone showed emotion. And that was important because we noted that there was definitely not the neurotypical way of expressing emotion, which I had predicted, but we wanted to know where that was occurring. Is it in the type itself, in sadness or happiness? Is it in the intensity in which they're showing that emotion? Is it in the duration, how long they're broadcasting it, or were they showing something completely unique? And then we took it one step further and really wanted to hone in and make sure that there was a difference. So if you'll see here, happiness, this is the autism, this is the neurotypical. The neurotypical showed happiness and sadness a lot more than the autism participants. But when we went to unique happiness and unique sadness, so these unique expressions that deviated from normal, but they were something different that didn't fall in one of these categories, we noted what it was. And as you can see, and I thought this was interesting, the neurotypical individuals were not as unique in their sadness, but they showed some levels of unique happiness that weren't neurotypical. So in the neurotypical group itself, we noticed some variations, but as you can see, much higher results on the unique expressions. Specifically, I noted that individuals with autism showed emotion restricted to the lower part of their face. And interestingly, they moved their mouth to the left-hand side. So when they were talking, they would kind of do this a lot, or their nose would twitch to the left. And it was really in this area that they were showing that emotion. And it was happening for sadness or happiness. They didn't show a lot up above. Sometimes their smile reached their eyes in a neurotypical way. But for the most part, they were just showing emotion on the bottom part of the face, which I thought was interesting. When we looked at micro expressions, individuals with autism were much more likely to be showing these micro expressions. So very, very brief emotions, very, very quick. Then they had a significant level of brief, half second to 1 second, emotions, which is slightly quicker, and not very many macro expressions, but neither did the neurotypical people. Mostly neurotypical people showed brief expressions, just a half second to a second. We went one step further and looked at the perception of emotion. So I wanted to see if there was a difference just in the muscles in the face, or if what they were thinking they were broadcasting was completely different. And interestingly, the perception of autism was way low. So I did a consistent and inconsistent measure. If they were consistent with what happiness was and they said only positive things, then they were rated as consistent. Whereas if they were saying, I don't know, or I'm happy, but also kind of sad, those would be rated as inconsistent. So as you can see, they were mostly inconsistent in their findings of emotion and their perception of it, whereas a lot of neurotypical people were very consistent. They felt something and they said that they felt the same thing that they broadcast. So opportunities in the future, this was super interesting. We had a pretty small sample size. In the future, I would want to see this done at a much larger scale, obviously. I wanted to be able to, in the future, use neuroimaging to repeat the studies. So do the exact same thing. They see images, they express how they feel, and someone records their face, but it's in a neuroimaging environment. We're able to use data from the brain to see which areas of the brain light up and see if it correlates or we see a pattern. Perfected National Security Agency protocols. There's a lot of algorithms in the TSA for the airports that flag people who show these signs of microexpressions because they've been used for deception. However, as even my research showed, in a very limited way, individuals with autism show a lot of microexpressions, and that is not necessarily from deception. In fact, I would argue that it is not at all, especially based on their responses to the questions. That's just how they were displaying those emotions. So this could help potentially help people that are autistic not run across law enforcement in a negative way. This could be used to branch out to test any further patterns other than happiness and sadness, to check and see if some of these are happening across all the other level emotions. And there's a greater chance of using iMotion's computer analysis. It's an expensive system, but it is a computer looking at their face so that there's not that human error that could take place. I did have Yaz Tolliver. She was awesome and did my second rating system for the coding so that there was inter-rater reliability. We were at a 96% agreement rate, and we looked into it and we were able to get to 100% agreement rate on these slides. So thank you so much for listening. I look forward, if you have any questions, you're welcome to email me. Here's my email. And thank you for letting me share my research with you. And here are my resources.